Welcome to our Unit A review. So we did a lot in Unit A, hours and hours of work. So I'm going to try and condense this as much as possible. There are about 29, 30 slides in this presentation. The last time I recorded this, I forgot to put my mic on and it took an hour. So I'm really hoping this is much shorter than that. I'm going to be referring you back to your lessons and the other recordings for concepts that I don't have time to go fully into. So by all means, this isn't the only review you should be doing. Make sure you get into those review packages and do some more. So we're going to be looking at Chapter 1, Circulation and Immunity, and then Chapter 2 is the Genetics unit. So to start off, each slide of this review package has the outcome at the top of the page that you need to make sure you understand, and then some information to help you understand that. So the first outcome is to describe the rhythmic contraction of the heart and its function in the general circulation of blood through the pulmonary and systemic pathways. All right, so when we're talking about rhythmic contractions, we're talking about how the atria contract, the top of the heart, and then the ventricles at the bottom will contract. So when the ventricles contract, that's called our systole or systolic blood pressure is created then. And then that blood from the, so the blood from the ventricles, when they're being contracted, it's going out of the heart. So either to the lungs, if it's on the right side, or out of the heart on the left side to the body. When the ventricles are relaxed, that is when they're filling with blood because the atria, the chambers on the top, are contracting and pushing the blood down. So both sides of the heart contract at the exact same time. So top, bottom to get your heartbeat. Also, we want to understand how the pulmonary and systemic pathways work together. So you have your heart split in two sides. The right side, remember it's opposite. So this is my right hand. Oh, well, not opposite in the picture in the video that you're watching. But anyway, so if you're facing you are in that screen and they're looking at your heart, you're looking at the opposite side. So the right side is on our hands left. Anyways, so the blood from the body is coming in. So it's in blue because it's low in oxygen. So we have to send it to the lungs. So the blood in the heart on the right side has low oxygen. All right, so it's going to the lungs and then it's coming back from the lungs into the left side of the heart, and then the left ventricle is going to pump all that oxygenated blood to our body. So the heart is separated in two sides that way. And then we've got the pulmonary circulation here, which we just talked about when that's going to the lungs. This is to get oxygen, to increase the oxygen, and to get rid of the carbon dioxide. And then once we've got high oxygen, that's when we want to send it to the body. We want to send our high oxygenated blood to the body, and then the blood comes back with low oxygen, high carbon dioxide to expel into the lungs. All right, the next outcome is to describe the principal structures and associated blood vessels in the heart. So the outcome is very specific and it lists the parts of the heart that you do need to know. So the ventricle, the atria, the septum, the valves, you do not need to know the specific names for them. Aorta, vena cava, and that says pulmonary arteries and veins, and then the coronary arteries. Remember, the coronary artery is on the outside of the heart. It feeds the actual heart muscles. So it's not being pumped through the heart. So anatomy of the heart, you've got your superior and vena cavas collecting all the deoxygenated blood, that's why it's blue, from the body, going into the right atrium. When the right atrium and left atrium contract, it's being pushed through a valve. So there is a valve there between the atrium and ventricles into the right ventricle. When the ventricles contract, that blood is pushed through another valve. So there is another valve between the ventricle and the artery. And this is the pulmonary artery. Pulmonary because it's going to the vein, and pulmonary because it's going to the lungs, right? And artery because it's moving away from the heart. So this is the only artery that contains low oxygen. The only artery. All the other arteries are high oxygen levels. All right, so it's going to the lungs, that blood, our lungs, to get rid of the carbon dioxide and pick up the oxygen, and it's going to come back through the pulmonary veins. Remember, veins are going to the heart. These are the only veins that are high oxygen. Only veins. All other veins are collecting blood from around the body that has low oxygen. Okay, so the pulmonary veins bring that blood back to the left atrium, and then that blood is pumped when the atrium contracts into the left ventricle through another valve, and then up through the aorta through yet another valve here. The aorta then takes that blood to all the rest of the parts of the body, head, toes, everything. So just make sure you know where the oxygenated and deoxygenated blood is flowing through the heart. It doesn't matter whether the pathway starts at the left atrium or, the vena cava. You need to be able to trace the path all the way back to where you started. So here's an example question from a previous diploma. So some blood vessels in the human circulatory system are listed here, and it says if a flu vaccination is administered into the arm muscle, so we're starting at the arm muscle, and diffuses into the blood capillaries, we're in the capillaries now, then the pathway of the vaccine once it enters a capillary is numbered. Okay, down below. So if you're in a capillary in your arm, that blood is picking up anything like this vaccine and giving off carbon dioxide, oxygen to all your cells, right? So from the capillary, the blood with the vaccine in it is going to move into venules, then veins, and then it's making its way through the main vein, the vena cava, into your heart. So you're going to be finding that vaccine in the vein in your arm first, and then it's going to hit the vena cava, and then the vena cava is taking that blood into the heart, into the right side, the right atrium, and then the right ventricle, which aren't listed here, but the right ventricle then takes blood to the lungs through the pulmonary, don't get it confused, artery, because it's moving away from the heart. So pulmonary artery is next, 4, and then goes to the lungs and comes back from the lungs in the pulmonary vein to the heart. So pulmonary vein is number 3. And if you hadn't noticed, the key is down below for most of these questions, not all, but a lot of them. All right, next outcome. Describe the structure and function of the blood vessels and the flow of blood through arteries, arterioles, venules, veins, and capillaries. So this actually would have been great before the last question because we were talking about being in a capillary at that point. So here is our flow of blood. We're going always high oxygen for the most part, other than the pulmonary artery, right? All arteries, other than that one, have high oxygen, low carbon dioxide. And as you move towards the capillaries, towards the veins, you're in the capillary beds, you're giving that oxygen to the cells, and you're taking up the carbon dioxide from the cells so that they can get rid of their waste, and then moving that through the vein. So make sure you know the oxygen content of the blood as it flows from artery to vein. Also the direction of the flow. Again, the blood is always flowing from the arteries to the veins. Make sure you understand the structure. So when we're looking at an artery versus a vein, artery walls are much thicker. You can see that in this picture here. Much thicker wall compared to this vein wall. Arteries have very elastic walls because they're under high pressure. When the heart beats, they expand and then they contract, so they add some pressure to also to that vessel. Veins have valves. Okay, only veins have the valves. Arteries do not, and the elastic walls are in the artery. And the most pressure is found in the arteries. Okay. There's a little picture down here of a capillary. Just a reminder that capillaries are basically just one cell thick. They can allow one red blood cell through at a time. This is so diffusion can happen easily. Remember, we did a whole lesson on blood vessels. So this is like a 2-minute summary. So you're going to want to go back to that lesson and make sure you review all the pieces around blood vessels. Here's an example question you might see on a diploma. The type of blood vessel depicted in this diagram is, immediately I am seeing the word valve and I see valves opening and shutting. Any blood vessel with a valve is a vein. Make sure you remember how to take blood pressure. Know what your systolic and diastolic readings are. The device used for blood pressure is sphygmomanometer. And know some of the factors that affect blood pressure and heart rate. Like if you're moving faster, exercising, your heart rate increases. If you're eating unhealthy foods, you can have narrowing of your arteries, so your blood pressure can go up. There's a whole bunch of factors that were in that lesson that you should review. Cardiovascular disease. So there's a few that you just want to make sure you understand. The section on cardiovascular disease. Again, there was a whole lesson on this, but this is just a little summary. We talked about varicose veins. This is when the valves in the veins weaken and the blood starts to pool down low because it can't get back up to the heart as efficiently. We've got atherosclerosis here, where you start to have this narrowing of your arteries, which increases the blood pressure because it makes it harder for the heart to pump blood through those arteries. If this atherosclerosis gets really bad, you can have a piece of that plaque that build up on the wall break free and block your artery. And if it's blocking the artery, the coronary artery, then like these arteries here, then the heart muscle isn't getting the oxygen it needs and it starts to die, and that's where you're getting a heart attack. The other example down here is a septal defect. We talked about this. Some babies can be born with this defect and has to be repaired. So this is when the septum here, it does not fully form. It should be going right across and blocking both of those ventricles, because you don't want this mixing of oxygenated and deoxygenated blood. Otherwise, if you do, it becomes a lot less efficient of a pump. So if your deoxygenated blood is pumping into the left ventricle, and this left ventricle is pumping all that blood to the aorta, that means it's pumping blood that's deoxygenated. It hasn't been to the lungs yet, and you really need it to go to the lungs first to get full of oxygen before you send it around to the body. Otherwise, your body is going to be lacking in oxygen. All right, those are just a few. Make sure you go back and review LDLs and HDLs and how they affect atherosclerosis and some other cardiovascular diseases that were listed in that lesson. All right, next outcome is where you're looking at the main components of blood. So you need to know plasma, red blood cells, white blood cells, platelets, blood proteins, and other antibodies, hemoglobin, hormones, anything that's in your blood. That it's you need to know that the blood's used to transport substances like nutrients, waste, gases, hormones. It's also responsible pieces in their blood for blood clotting and defense against pathogens and distribution of thermal energy. Not something we talked about a whole lot, but that is one of the jobs of the blood, is to keep our internal body temperature very similar. So it's bringing that heat all around our body all the time. So the different components of blood, if you take blood, put it in a centrifuge, it separates into different components, red blood cells being the heaviest at the bottom. My picture's blocking it, but about 45% of the blood is red blood cells. Then you have white blood cells and platelets above them, only about 1%, so a very small amount, less than 1% even. And then plasma makes up the majority of your blood, which is like a watery liquid substance, which is carrying any kind of nutrients or protein, hormones that you need to transfer around your body. Your red blood cells or erythrocytes, they're the ones that have that hemoglobin, which needs iron to form, and this is the compound that carries oxygen. The shape is that biconcave, like a donut without quite the hole in there, but both sides are rounded in. Your white blood cells are leukocytes, and they're specifically for fighting infections. This is not to scale. White blood cells tend to be much larger than the red blood cells in the pictures you've seen. And then platelets are like pieces of, they're all jaggedy shaped pieces of cells, thrombocytes. And then the plasma holds all the ions and the blood into suspension, and any other nutrients or chemicals that you're carrying along in your blood. There's an example question that you might see on a diploma or your unit test. Doctors and technicians at a medical clinic treat and counsel patients and carry out technical procedures during their daily routines. Blood clotting factors are routinely tested. Clotting is initiated by, well, anytime you see the word clotting, you should be automatically thinking about platelets. All right, so on to the immune system. This was the last lesson in our chapter 1. There are 3 outcomes here that we're going to look at first. The first one is describe how pathogens in the environment, how do they get into the circulatory system, and to create these adverse effects. Also, we're going to look at different body mechanisms, including the skin and body secretions that prevent pathogens from actually getting in. And then explain the relationship for autoimmune disease and the human immune system. Woo! Okay, first line of defense. So this is where we're talking about skin, hair, stomach, pH, eyelashes, all these things we have to keep out bacteria, viruses from even getting into our body. There was a great diagram about that described a whole bunch of these different first line of defenses. Diseases, how are they spread? Through different pathogens. Here are a bunch of different examples of pathogens. There's an example of a virus, AIDS, excuse me, parasites. There's a bacteria here that causes leprosy. And then autoimmune diseases. This is when your white blood cells go and attack your own body cells. And specifically with rheumatoid arthritis, they're attacking body cells that are in the joints, whether it's cartilage, bone, whatever, causing deformity, pain, and swelling, which are all signs of the immune reaction. So an example question you might see on a test, you have an example of a person's non-specific first line of defense against pathogens is tears. All right, so that non-specific and first line of defense, these are what we're talking about when we're talking about those physical defense barriers that our body has created. Now we're going to get into the specific immune response. And the outcome you can see describe in general terms how immunity to pathogens develops, how the immune system responds to foreign antigens, and the roles of, you've got macrophages, B cells, helper T cells, killer T cells, suppressor T cells, memory cells, and antibodies. So make sure you know all those different cells and what they do. I have reviewed and listed them here. This is my favorite diagram still of the immune response. Something about it, I just find it really nice and simplified compared to the textbook one, but feel free to use whichever one you like best. So step 1, your antigen enters the body. So antigen being the foreign invader. Okay, there's your antigen for an invader. So whether that's a bacteria or virus, whatever it might be, then your macrophage, there's your macrophage. It comes in, engulfs that antigen, the invader, and pushes the antigen markers to the surface of its cell. This lets the helper T cells come in and copy that shape. They're so helpful, and tell the B cells to start making antibodies to match that shape. So we're talking about T cells. I've listed all the different types of T cells here together. So there's our helper T cells. So they're helping get those B cells going. So the antibodies then go in and attach to the antigens. And then step 6, the macrophage either engulfs the antigen, or those killer T cells come in and start puncturing the membrane of the bacteria to death. All right, so we have our killer T cells, they puncture the membrane. And then our suppressor, whenever you see the word suppressor, you think stop. They stop the response, suppressor T cell, and then memory T cells are going to remember those antigens. So the next time they come in, they have those antibodies ready and are faster next time. And because if you're faster, you can help prevent the actual reaction to that pathogen. So B cells' main job here is the antibody production. So there might be memory B cells that remember how to make those antibodies, but antibodies is the big thing that B cells are going to be doing. All right. There's an example here of a previous diploma exam question. So pathogens have a unique substance on their surface called number I, and a type of white blood cell that uses these unique substances to recognize then engulf pathogens is 2. So pathogens we talked about, these are antigens, right? They have antigens on their surface, antigen markers. And if you're talking about engulfing a pathogen, then you're talking about a macrophage, because the killer T cells are going to puncture and to get rid of the antigens. So the answer is B in this case. All right, on to vaccines. The outcome, analyze how vaccines defend against disease-causing bacteria or viruses. So vaccines work by injecting a dead, synthetic, or weak pathogen into the blood so the antibodies can be developed. If you have an antibody already and you have an existing pathogen coming in, like measles, because you've had your measles vaccine, then your body can quickly make that antibody and protect you from this pathogen and kill all those pathogens and get rid of them before they can cause any damage in your body. If you do not have the antibody, that's when you are going to get the disease, or your immune response is going to work as hard as it can to get you so that you don't have the disease. So that's where vaccines are handy. They come in and they train your body ready to be prepared for the disease coming in. Alrighty. So next outcome. So this is where we start going into chapter 2, where we're talking genetics now. So in general, we're going to look at the behavior of chromosomes in this outcome during mitosis, meiosis, and fertilization. So first off, what are chromosomes? They are where they found. So the location of them, they are found in the cells, and more specifically in the nucleus of the cell. Chromosomes contain DNA that are all wrapped up, coiled up really tightly into these chromosome structures. There are 23 pairs or 46 individual chromosomes in a typical human cell. You can get genetic disorders where you have extra chromosomes. So if you have an extra chromosome 21, that's Down syndrome. Make sure you understand how to read this 23rd pair, the X and Y, or X and X chromosome. So X and Y, this gives you a male. And XX, if you have 2 X chromosomes, that's a female. And remember that Y chromosome is much shorter than the X chromosome. So if they're not labeled, you should be able to recognize them right away as a male, because that Y chromosome is so short. Typically, chromosomes come in pairs and look like the X. They've been duplicated, sorry, not pairs. So it's a duplicate, but one chromosome looks like this, and it's just been duplicated, and that's why it looks like that X form, because it's waiting to separate. So most of the time it's spent in that format, or it's depicted that way too in the karyotypes. All right, outcome. Describe in general the behavior of chromosomes during mitosis, meiosis, and fertilization. So same outcome as the last one. So now we're looking at mitosis and the chromosomes in mitosis and what they're doing. So first off, what is the difference between mitosis and meiosis? So mitosis, think toe, you stub your toe, oh, your toenail fell off, you need to regrow your toenail, your cells are going to do mitosis. So mitosis wants to create new cells that are identical to the cells that were already there. So for mitosis, you're going to have a diploid cell, so it has all the full complement of all your chromosomes, and you're going to form identical new diploid cells. So one cell replicates and divides into two cells, but the chromosomes are identical, right? You don't want to be creating cells that are different than the originals if you're just regrowing a toenail, right? They want to be identical. This occurs everywhere in the human body. All right. In meiosis, this is when you want to form a sperm or an egg cell, and you only want half of the number of chromosomes in your sperm and egg cells. So from one original cell, it's going to go through two divisions and form these haploid cells that have half of the amount of chromosomes in them. All right, this only happens in the ovaries and testes, and this is how we get genetic variation. So down here, when we've got fertilization happening, we've got the egg from the female, sperm from the male come together in a fertilized egg, and this is where the variation occurs. So we've got the mixing. So we've got one chromosome, or half the chromosomes coming from the egg, half from the sperm. They come together into that egg, fertilize, and you get the mixing of those different alleles. And how they're going to interact is the next few lessons. All right, so there's fertilization. So here you're going from haploid in the eggs and sperm and back to diploid, the full amount of chromosomes. And then from this point on, from the fertilized egg point on, it's going to be going through mitosis every time to grow bigger and bigger and bigger. The only time you do meiosis is to form sperm and egg cells. All right, here's an example here. You have a type of cell division happening, and if I look at this first cell division here, I see 1, 2, 3, 4 chromosomes. Right here, the 4 chromosomes have duplicated so that they have a partner, and then those partners are pulled apart, and then these new cells, you see, they still have 4 chromosomes in each one. All right, this is called 2N. It's diploid. And that's the original cell is always going to be diploid. Okay, original cell is always going to be diploid in these mitosis or meiosis. And you notice the same number of chromosomes in this last cell, so they're still diploid. Right? So cells are either diploid or they're haploid, half the number of chromosomes. The only ones that are haploid are eggs and sperm. All right, so don't let the 4N confuse you. That means nothing. All right, it's either 2N or N. So, and these cells are going from 2N to 2N. So diploid to diploid. So C is your answer in this case. All right, next outcome. Distinguish autosomal from sex-linked patterns of inheritance. So first off, for patterns of inheritance, there's a lot of different terms that you've been using in this chapter that you want to make sure you understand. So here's a quick review of some of them. So we have dominant and recessive genes. When there is a dominant gene present, it's going to overpower any recessive genes that are present. So dominant is also represented by capital letters. Here we have 2 dominant alleles, which provide a purple flower. Here we have 1 dominant and 1 recessive allele. We still get the purple flower because the dominant is overpowering that recessive allele. The only way we're going to get to see what the recessive trait looks like is if we have two recessive alleles, and then we get the white flower. You are called, or an organism is considered a carrier if it has one of the recessive alleles, but it's got the dominant allele still, and so the trait that it expresses or shows up is the dominant trait, but it's still got that recessive allele and it's carrying it. Alleles, what are alleles? I keep using that term. There are different variations for a trait. So in this example here, you have the purple allele and you have the white allele for flower color. Homozygous means, homo means the same. So here you have homozygous dominant because you have two dominant alleles. Homo, same. Two. Here heterozygous because you have different alleles. So one dominant, one recessive. So when you say heterozygous, you don't have to say heterozygous dominant or heterozygous recessive because you have to have one dominant and one recessive allele. If you're talking about something that's homozygous, you do need to say, is it homozygous recessive or homozygous dominant, right? Genotype versus phenotype. There's a good explanation over here on genotype versus phenotype. Genotype is the alleles, description of the actual alleles. So it could be, these ones are homozygous dominant or too big A. And the phenotype is what is expressed. So how does that trait look and how is it expressed in the moth? It's expressed as a dark color, right? Genotype, phenotype. Punnett squares. We use Punnett squares to help predict what the offspring are going to look like. It just gives us a prediction. There's no guarantees, just an approximation, right? So let's take a look. This is the autosomal pattern of inheritance, whereas over here is a sex-linked pattern. Okay. So in this case, yellow pea pod is the dominant allele. So this parent here is heterozygous. This parent here is homozygous recessive. You do your box where you're moving the alleles around, seeing how they're going to recombine. What are the recombination possibilities? And in this case, half of the offspring, so 2 out of 4 offspring, or 50% of the offspring, are going to be homozygous recessive and have the green pea pod, whereas the other half, 50%, odds are that your other half of the offsprings are carriers of the recessive gene, but they're going to have yellow pods because they're heterozygous. For sex-linked traits are always showed on the X or Y chromosome. And typically in this course, you're only going to see the traits on the X chromosome. So the Y, you'll notice, does not actually have an allele for this. It's partly because the Y chromosome is so short, it's missing a lot of different alleles that it could have. So it's a lot of these different traits are determined by that X chromosome. And the X chromosome is coming from the mother. Well, no, that's not true. The father's, no, ignore that last statement. The father can give either the X or the Y chromosome. All right, so if you have a carrier mother here, that means that she has the allele for the disease that we're studying, hemophilia, which is a recessive allele. But she also has a dominant allele that keeps her healthy so she doesn't get the disease. You have an unaffected father, so the father does not have hemophilia, so his 1X chromosome is a healthy chromosome and it's dominant, healthy, and he has the Y chromosome. So all his sons are just going to get his Y chromosome. So these are all the sons here along the bottom. Sons. These are all the daughters because the man has given his X chromosomes. If the father donates an X chromosome, you get a female. You get a daughter. If the father donates the Y chromosome, you get a son. The mother, the mother's, in this case, the mother's chromosome here is going to determine if the child has hemophilia. That's the only hemophilia allele here. And in this case, this daughter here is going to be a carrier, but she won't have the disease because her father gave her the dominant healthy gene. But this son here got the Y from the father, and only the X chromosome that has the defect, hemophilia defect in it. So this son is going to have hemophilia. So if the question is, what percent chance that a son has hemophilia, it's going to be 50-50, right? So there's only two possibilities for the sons, these two. 50% chance that a son will have hemophilia. None of the daughters will have hemophilia, but 50% of the daughters are likely to be carriers. If you look at all of the offspring, if you just say all the offspring, not son, daughter, all of the offspring, 25% chance that one of the offspring will have hemophilia. Just this one here. Okay, so it depends. You got to look at the wording of questions, whether you're talking about sons, daughters, or all offspring. Watch those question wordings. Okay, here's an example question. Albinism, which is the lack of pigmentation in skin, hair, and eyes, is determined by a recessive allele, A. All right. Normal pigmentation is governed by a dominant allele. So this is normal. All right. So we have so we have possible genotype combination of parents. So we want to know the sets of parents listed above that could produce a child who has normal pigmentation. Okay, so normal pigmentation would be a child that has a dominant allele. It has to at least have one dominant allele. It could have two dominant alleles, or it could have a dominant and a recessive, and it would still be normal. It just can't have that. That gives albinism. All right, so the first question, we have two homozygous dominant parents. So both parents, I'm just going to pop them right into a Punnett square, are homozygous dominant. So there's our dominant parents. Okay, this is obvious. We're going to see this, and we're going to see every single offspring is going to be also homozygous dominant. So yes, the first one, for sure, you can have a child that has normal pigmentation. It just says a child. It doesn't have to be all the children. So the next question, two homozygous recessive parents. Okay, so we've got two homozygous, means same, and recessive parents like that. So all these kids, again, are going to be homozygous recessive. Not a single kid without albinism in that family. So not number two. So we don't have any normal pigmentation there. One heterozygous parent is the next question. One heterozygous parent and one homozygous dominant. Homozygous dominant. All right, yes. So every child that this parents have are going to have a dominant allele. So they'll all be normal pigmentation. So 3 works, 1 works. Oh, I already put 1 in there. For 1 heterozygous parent and 1 homozygous recessive. So heterozygous and then homozygous recessive. Homo means same and recessive like that. So because there's a dominant allele here, we definitely will have a child with normal pigmentation. Okay, so 4 is true. And then 2 heterozygous parents, they're all like this. As long as you have 1 dominant allele showing up in a parent, you are going to have a child that has normal pigmentation. All right, so there's 3 of the kids that 3 out of 4 chance that the offspring will have normal pigmentation, and only 25% chance that they'll be have albinism. All right, so it's really only the number 2 that couldn't produce offspring that had normal pigmentation. All right, another example. Which of the following crosses would provide the greatest probability for albinism in the offspring? All right, so greatest probability for albinism. Remember, albinism only occurs if you've got that. So you'd think more of those recessive alleles, the better your odds of albinism. And you'd be right. Okay, so C is the case scenario here that will give you the most albinism offspring, but not a bad idea to try testing this out with all of them. I'm just going to do these ones to see. All right, so 50% of the offspring are going to have albinism. You can test the other ones to see what they would be. There was another example question after this one in the slides, so make sure you go and do that. I had to remove it from the presentation because I didn't have enough space at this time. So I'm just going to move that up here. So on this page, we're looking at DNA. There's 4 outcomes here that are going to be covered on this slide. Again, this was a whole lesson, so go back and check this lesson out. Describe the structure of DNA by identifying the structure of DNA as a double helix. So we talked about the double helix. There's the shape here. We could talk about the sugar phosphate backbone. So going around, those are the sides if you're holding onto the ladder. And if your feet are on the ladder, those are the base pairs. And those base pairs here, this is where it's unraveled, they're being held together by hydrogen bonds. The base pairs align up with partners, and the partners are always hydrogen bonding with the same partners all the time. You have your 4 nitrogen bases, and these are the partners. A and T and C and G are always lined up. So cytosine is C, guanine is G, adenine is A, and thymine is T. Review the people who were instrumental in the discovery of DNA structure. And then also replication. So complementary DNA strands. So over here, during DNA replication, the double helix will unzip. So replication, why do you need to do this? Well, when you need to make copies of like your chromosomes for, say, mitosis or meiosis, you need to replicate your DNA. So that awesome structure, the double helix unzips, new nucleotides come in, match up, pair up. They always pair up in that same way, A with T, C with G. And then you can get 2 new complementary strands that are identical to the original. So if one strand is down here, we're just looking at this example, if one strand is CGATTA, the complementary strand would be G, because C and G always bond, and then C, and then T, and then A, T. If you're not sure how I did that, make sure you go back to that DNA lesson to figure out complementary strands. You'll definitely need to be able to do that. Alrighty, on to the next part here. So, why DNA? What is it doing? What DNA is doing is it's holding the code for all these genes. And what are genes? They're proteins. So proteins rule our body. They provide our structure, energy, enzymes to regulate every chemical, every reaction that's occurring in our body, transport things around, help us move, give us immunity, hormones, sensory, all these different things are run by proteins, and the code for these proteins in our body are held within our DNA. So here are outcomes. Describe the primary function of DNA by describing how amino acid sequences of a polypeptide, which is the same thing as a protein, is determined by the sequence of the DNA triplet codes. So we have to use the table on page 13 here to match those DNA triplets to find the amino acids that make up a protein. And then describe the role of proteins in the human body as regulatory molecules, so like enzymes, structural molecules, and sources of energy. And then the last outcome, mutations. That one's we cover in the next slide. You can ignore that one for now. Okay, so proteins, like it mentioned in the outcome, they're made up of amino acids, a whole bunch of different amino acids in any kind of order, in a big long chain. There are only 20 amino acids you could choose from to make up this chain, but those 20 amino acids in different orders, different lengths, code for every protein that our body needs. Quite amazing, really, when you start to think about it. And how do we get this code? Well, our DNA triplet, so 3 DNA nucleotide bases, will code for 1 amino acid. So say you have your DNA strand of C, T, G, A, T, C, something like that. That was a G. You're going to split this DNA sequence into 3 nucleotides at a time, and that is going to code for an amino acid. So we're going to use our table. We'll do one together. We'll look up the first base. It says first base here is C, and there is my C. And then T is the second base, so there it is up top. And then G is hidden by my face. So there's our G, our third one. So down here, can't exactly see what it says, but I think it says leucine. I think it says leucine. So that's your first amino acid that's being coded for. Make sure you have your data booklet available when you're doing your unit test. You will need that. There's an example question. The key is down below here. Make sure you try this with your data booklet. If you want to pause right now and give that a go. It's too hard for me to flip around with the data booklet, so I'm not going to do this one. That's why I did an example on the last page. All right, mutations. Describe how mutations in DNA affect proteins, resulting in human diseases. And I think the second outcome we do on the next slide too. It needs to be moved. Okay, so make sure you know some mutagen examples, things that cause mutations in the environment. So environmental things that cause mutations, like smoking, there's a whole list of them, radiation, x-rays, that kind of stuff. Make sure you know those. There's also mutations that occur just spontaneously too. You can have chromosome mutations. That's when you have an extra chromosome because of the things didn't divide properly, cells didn't divide properly, especially during meiosis. So you might have an egg with an extra trisome, extra chromosome. Okay, then there are these point mutation and frame shift mutation. So a point mutation is when you have a single nucleotide that's changed, just one. So if you take the sentence time to dream and you change one letter from the I to an A, you get team to dream. So one of those amino acids is going to get changed. It may cause a major issue, or it may be fine and the rest of the protein is still intact and it's okay. So that's a point mutation. The frame shift mutation is when you add or delete. So you could insert an extra letter and that just messes up everything. So the frame has been shifted now so that the everything after that point doesn't make sense. All is going to be different. Big issues there. Same with the deletion. Anything after the deletion, any amino acids after the deletion will all be messed up because you've shifted the frame of the 3 nucleotide code to be read. Make sure if you don't understand what's going on, you go back. This was a whole lesson on mutations as well. So bacteria, antibiotic resistance. So bacteria can, when it's replicating, it can spontaneously get these mutations just like humans, and bacteria is replicating a lot faster, so their mutations can happen a lot quicker. One of the mutations they could get is antibiotic resistance. If say you're taking an antibiotic and you've got bacteria around, that bacteria that spontaneously gets that resistance could start living and thriving and start reproducing. When bacteria meet other bacteria or hang around the environment, they actually will throw out pieces of DNA or transfer plasmids. So make sure you look at transformation and conjugation. So bacteria don't actually have to replicate with each other to get variation. There's a lot of ways that they get new variations and share their DNA. They're very sneaky that way. So take a look at that. We've got some genetic diseases like sickle cell anemia and hemophilia, which are caused just by, one of them is caused by a point mutation only in one of the proteins that's involved. Make sure you go and review those. And keep in mind too that there are beneficial mutations. There are mutations that help organisms survive better, right? They create that variation in our population. That cheetah that could run much faster than the other cheetahs had a better chance of surviving and mating and passing on its genes. That ability to run faster might have been due to a mutation. So a lot of evolution and changes and variation in our population are due to mutations. So don't just think of mutations as a bad thing. Think of them as it's a constant evolution and variety and creation of variety in our world. All right, outcome. Investigate with the aid of a pedigree chart the familial inheritance of a specific trait that is controlled by a single pair of genes. All right, so when you see these pedigree charts, remember that if they're darkened in, that means they're affected. If they're a square, they're male. If it's a circle, it's a female. You can have to use these charts to figure out some genotypes sometimes. If you start to notice there's a lot more men or only men that have a gene that is happening, like a trait that's being represented in a pedigree, that could mean that it's a sex-linked trait. There's a question here, example here. Okay, so now this is red-green color blindness pedigree. So a doctor used the color blindness pedigree above to trace red-green color blindness in a family. According to the pedigree, how many women in the family are colorblind? So everybody's a square except this woman here. So there's only one. All right. And then the next question is, okay, so that's also a hint too, right? Color blindness might be a sex-linked trait because there's only one woman with the trait. All right. So the genotype and phenotype of, but it might not be either. So it didn't tell us. So we don't have to jump to any conclusions. The genotype and phenotype of 2-5, this woman here, the mom of these kids, is respectively. All right. So the mom does not have color blindness. However, she had to have passed it on. So she must be a carrier, right? So she has to be a carrier because these offspring must have both recessive genes or recessive genes for the color blindness, whether it's sex-linked or not sex-linked. She must be a carrier. If you're a carrier, then you're heterozygous, right? She still has one of the dominant genes that makes her not colorblind. And she's not colorblind. So D is your answer to that. I'm just going to go back for a sec. When you're looking at these, the trick is if they're asking you about a parent's genotype or phenotype, you have to look at the offspring. What's being produced. That's especially a parent like this one is not from these parents up here. She did not come from them. She married into the family. So you have nothing above to look at. You need to look at the offspring that she has produced to see what combinations could be possible. All right, outcome. Describe in general terms genetic engineering and its application to gene therapy and the development of genetically modified organisms. So there was a question in one of your practice questions in your textbook asking you why selective breeding is considered a genetic engineering. It's because you're choosing those useful characteristics and purposefully breeding to get more of those useful characteristics. Make sure you take a look at the corn, where it came from, and how it was selectively bred to become corn from this looking wheat-y looking thing. lastly, last page here. So the outcomes for genetic technology, in general terms, genetic engineering and application to gene therapy and the development of genetically modified organisms. And the next outcome, to explain the decisions regarding the application of scientific and technological developments involving a variety of perspectives, social, cultural, environmental, I can't see another one, and economic considerations. Look at the risks and benefits. So there's a whole page on the risks and benefits too of genetic technologies. Make sure you take a look at that. Some of the genetic technologies to review DNA fingerprinting, how it's used for paternity tests or identifying criminals in cases. Transgenics, this is where you're mixing two different organisms' DNA together to create, or adding more to create an organism that has characteristics that you want it to have. Recombinant DNA, you would use that in GMOs, but you can also use it here in this example where we're taking a bacteria plasmid, popping in the gene for human growth hormone into that plasmid, and then putting it back into the bacteria. And then the bacteria will grow and create and produce this growth hormone that we can keep in a lab, and then have that growth hormone for humans that need that growth hormone. You can do the same thing with insulin. You can get these bacteria creating insulin for you. Get them creating whatever protein you need for medical purposes. Then there is gene therapy, and this is where you're actually going into the human genome and changing the actual structure of the DNA inside a human cells, so if there's a defect in there, you're going to go and change that gene. There's a list of risks and benefits of some of these technologies. Make sure you review that. And then the last slide in the very last lesson was on the perspectives, and that's these different perspectives. And in the actual slide presentation for this review, there are 3 more slides with some questions on genetic technologies and some of these perspective questions. Make sure you review those. The answers are down below on each slide. If you have any problems, contact me. Awesome. All right. Have a great day and good luck on your unit test.